Hey everybody, welcome back to another AP Biology video. In this video, we're going to cover the topic of DNA replication, which is the process by which we create a copy of the DNA molecule that exists within the nucleus. So, let's look at a few things that we should remember from previous topics that will help us understand this process. First, we should recognize that DNA consists of two strands, complementary strands, of nucleotide monomers, and that when we look at the structure of these monomers, we should recognize that we have a phosphate group that's attached to the 5' carbon on our pentose sugar, and a hydroxyl group attached to the 3' end of our pentose sugar. This is going to be important because the entire process of DNA replication is a very direction-focused process. So we are always going to be adding nucleotides to our existing strand in a specific direction. So keep these carbons in mind when they come to the structure of the nucleotide in order to help you identify which direction we're moving in. Now, we talked about the process of both mitosis and meiosis in previous units, but we should remember that DNA is replicated or copied during the synthesis phase of interphase. So remember that interphase is composed of three subphases. We have G1, S, and G2. So it's during this S phase, the synthesis, that we are copying the DNA so that we have an identical set of DNA to pass on to each of our daughter cells. Now, we won't really get too far into any of the other processes today, so we are just looking at what is happening during synthesis. One of the unique things about the process of DNA replication is that this is what we would call a semi-conservative process. So what does this term mean? So when we say that something is conservative, that means that everything is conserved, that we're keeping the same things from before. Well, when we have something that is semi-conservative, we're keeping parts of the original and creating new parts to go along with those. So in this process, we have these original parental strands of DNA that we're going to use as templates. So we can read the parental strands and use those strands to create our new strands, what we call daughter strands. So each one of our replicated DNA molecules that we get out of this process is actually going to consist of one parental strand and one daughter strand. So if we look at the process of DNA replication over a few generations, we can see that each time we replicate, we retain a part of that original DNA molecule, the part that we used as a template, and we build a new strand. So this is in essence the depiction of our semi-conservative process. Now, when we get into a process like replication, we need to know where to start. So DNA replication begins at a specific sequence along the DNA molecule that we call the origin of replication, which we often shorten to ORI because origin of replication tends to be a little bit of a mouthful when you're saying it quite often. Remember that on our prokaryotic cells, our DNA sequences are relatively short and they are composed in these circular chromosomes. So they are only going to have a single ORI. And replication is going to proceed in both directions away from that origin point. we can compare this to something like eukaryotic cells that have so much more DNA within their cells, and we have these linear sequences. So this process would take forever if we only had a single point on each of our DNA molecules. So in eukaryotic cells, we have these larger chromosomes, and therefore they have multiple Ori sites. So each time that DNA replication is going to proceed away from these replication sites, and the replicated DNA will actually eventually meet another sequence of replicated DNA. So we will have to put these sequences together in order for us to have our final product. Now, really, the main focus of a process like DNA replication is going to be in the enzymes that make it possible. So DNA replication is facilitated by several important proteins and enzymes with specific functions. Now, like most things that we discuss in AP Bio, we can always go deeper, but I'm going to focus on the ones that I want you to remember for their functions. So we have one of our key players is helicase. Helicase has the function to unwind the DNA helix. So remember that we have these two strands that are attached to each other by hydrogen bonds between their nitrogenous bases. And the reason that we have these hydrogen bonds is because they're fairly easy to break and also fairly easy to put back together. So helicase has the job of breaking the hydrogen bonds that exist between these complementary nitrogenous base pairs so that we can separate our DNA strands. And this process often looks kind of like an unzipping when we look at it in models. In order for our DNA molecule to not completely twist up on ourselves, or what we call supercoiling, we need an enzyme known as topoisomerase. So topoisomerase actually will cut a sequence of DNA and then will rejoin it prior to leaving in order to relieve the strain that is caused by our DNA polymerase molecule. So this eases the process of supercoiling. Now, in eukaryotic cells, topoisomerase is the enzyme that does this, but you may see it presented in other models as an enzyme called gyrase, which is what we see in prokaryotes. Now, once we have our DNA strands separated, they're going to want to come back together. So we need something to hold them apart. And this is where we get these single-stranded binding proteins, or SSBs. These prevent the reannealing or the reformation of those hydrogen bonds in order to keep our DNA strand separated. So these start the process of DNA replication, but the actual replicating is completed by an enzyme sequence that starts with primase. So in order for us to know where to begin DNA replication, we need a starting point. And this starting point is indicated by a sequence of RNA primers. And these will be replaced later on before we get our finished DNA molecule. But these are essentially the green light for where DNA polymerase will need to begin. So our heavy hitter here is DNA polymerase 3. And this is the enzyme that synthesizes or makes our new DNA strand by taking nucleotides from the environment and attaching them via hydrogen bonds to our template strand according to these complementary base rules. So remember that if we have an A on our template, we get a T. If we have a C on our template, we get a G, and vice versa. Now, I mentioned that we get these sequences of DNA that need to be connected together. So the final enzyme that I would like to highlight is that of ligase. Ligase functions to kind of glue different segments of DNA together. And this is equally important for when it comes to our lagging strand, which we'll talk about, which is created or composed of these Okazaki fragments due to the directionality of our DNA polymerase III. So if you guys are good, let's go ahead and jump into this process and see it happen step by step. For our first stage, topoisomerase is going to bind to our origin of replication and ease the strain that is created due to the supercoiling or the twisting of our DNA double helix. From here, our helicase is going to come in and break the DNA bonds, the hydrogen bonds between our DNA strands, and separate those sequences. And you can see here that at the end of our sequence, we have these single-stranded binding proteins that attach to the separated strands and keep that replication fork open. So next, once we have these separated strands, primase is going to come in and lay down an RNA primer. So this indicates where replication needs to begin as DNA polymerase III gets in to do its work. So starting at that primer, DNA polymerase III is going to come in and add new nucleotide bases complementary to those of the template strand. It's important to note here that DNA polymerase III only works in the 5' to 3' direction. Okay, this is important. Remember the direction that DNA polymerase III is going to move in, because that creates differences between what we call our leading strand and our lagging strand. Now, once DNA polymerase III has completed the strand, DNA polymerase I is going to come in and replace our primers with a DNA nucleotide. So we don't want to have these RNA primers left into our strand because that'll throw everything off. And once we have replaced our primers, DNA ligase, which is our final enzyme, comes in and it binds together these segments so that we have a full continuous sequence of DNA. So let's take a moment to look at some of these differences between our two strands of DNA. So once we separate into this replication fork, we create two versions of single-stranded DNA molecules. And the one that proceeds in the appropriate direction for DNA polymerase III to do its job, and in the same direction of our replication fork formation, is our leading strand. Our leading strand is notable because it exhibits continuous replication in this 5' to 3' direction. And again, this is in the same direction as our replication fork movement. So it's often easy to identify which is our leading strand because of these two key characteristics. This strand only needs the one RNA primer for DNA polymerase to get started and then create the daughter strand from the template. Now, if we compare this to our lagging strand, things get a little bit more complicated. Our lagging strand has to be made in this discontinuous fashion because it is not in the same direction that DNA polymerase III likes to read. Remember, with our complementary bases and our two strands, we have these anti-parallel directions when it comes to the sequence. So the lagging strand is still replicated in a 5' to 3' direction, but DNA polymerase kind of gets a little flustered when it has to work in conditions that it's not set up for. So what we do to get around this is we set up multiple RNA primers that can then be used as individual starting points that can be connected together later. So these small segments of DNA that are formed via this process are what we call Okazaki fragments, named after the researchers who discovered them. These Okazaki fragments are eventually annealed or connected by that DNA ligase to create a continuous strand. But we have to create these fragments first and not just have the nice single strand like we get with our leading strand. So let's look at that process put all together. Now, DNA replication as a process is not perfect. In fact, often we find that errors can be made. Even the best of us can make errors from time to time, even DNA polymerase III. So at this point, these mutations can occur at about a ratio of 1 to every 100,000 base pairs, which is very often when you consider how many bases exist within our DNA. So we can divide our mutations into categories based on how they affect the original integrity of our strand. So we have three different cases here. We could have a substitution where we swap in the wrong nucleotide for the template version. We could have an insertion where we insert a bonus DNA nucleotide. I'm sorry, I am just realizing in my animation here that I put the insertion in the wrong spot. That, what we just saw, was a deletion where we removed a nucleotide from our strand and therefore had to shift everything over. That means that this next one will be the opposite as well. I will fix this in post. So deletion was what we just saw, but this was an insertion here. So we've added a nucleotide to our strand and then shifted down in the direction. So we have proofreading mechanisms to make sure that we experience as few mutations as possible, and that is where another DNA polymerase comes into play. DNA polymerase II is our proofreader. So what it can do is it can go down the nucleotide sequence and it can identify, hey, where are there errors? And so it can fix those mistakes. Now, this can reduce our error rate to about 1 in 1 billion base pairs, so much better overall considering the amount of nucleotides we have to work with. We have other repair enzymes that can fix errors not fixed by polymerase II, and there's all sorts of different processes that your DNA sequences can go through in order to fix the problem at hand. But we will just leave it here with the basics. Now, an important sequence that we want to consider when we look at our overall chromosomes is these non-coding regions that are there to essentially protect the important bits. So these are what we call telomeres. And telomeres are different from the coding regions of our chromosomes because they don't code for anything. They're non-coding regions. These are like the caps on your shoelaces. They prevent everything from getting damaged as you are wearing and tying your shoes every day. So every time that we go through a process of DNA replication, we can damage the DNA that exists. So we get over time some shortening of our telomeres. Every time that we replicate our DNA, we get less and less of the telomeres present in our product. So this happens naturally over a period of time, and this is one of the ways that our cells can identify, hey, it's time to stop this process. Because overall, once we've shortened our telomeres to a significant enough level, we don't want to start damaging the actual important DNA. So it's best to stop, and then our cell can undergo our apoptosis to break it down and or create new cells. This is a significant factor of aging that we see in many species. So this was a brief overview of topic 6.2 on the topic of DNA replication. And we will look at the next part in our central dogma in the next video. And I can't wait to see you guys then. Catch you later.